Welcome to the Journal Club, a new feature for the Society of Ethnobotany, formerly Economic Botany. The idea is to share current research, especially articles that appear in our own society's journals, but other books and book chapters and articles can be nominated, of course. We plan to meet online quarterly for about an hour and a half. I'd like to thank our host, Dr. Ethan Russo, for presenting today, to Trish Flaster and Amelia Neely of the SEB, as well as the SEB board for this journal club concept. Dr. Ethan Russo, MD, is a 1973 graduate of the University of Pennsylvania in psychology, University of Massachusetts Medical School, 1978, and trained in pediatrics and pediatric and adult neurology at the University of Washington. He was a clinical neurologist in Missoula, Montana for 20 years in a practice with strong chronic pain component. In 1995, he pursued a 3-month sabbatical doing ethnobotanical research with the Machiguenga people in Parque Nacional de Manu, Peru. He has held faculty appointments as an adjunct associate professor, Department of Pharmaceutical Sciences, University of Montana, and clinical assistant professor at the University of Washington School of Medicine. He's author or editor of 7 books and has published over 70 peer-reviewed articles. As I said, he's a board-certified child and adult neurologist, a psychopharmacologist, and founder and CEO of Credo Science. He is also Chief Medical Officer to Andira Pharmaceuticals, Senior Medical Advisor to Canurta, and Medical Director of Breeders' Best. He's Director of Research and Development for the International Cannabis and Cannabinoids Institute, 2017 to 19, Medical Director of Phytex, 2015 to 2017, and from 2003 to 14, Senior Medical Advisor and Medical Monitor Study Physician to GW Pharmaceuticals for numerous 1 to 3 phase clinical trials of Sativex and Epidiolex. Our first meeting features Dr. Russo's recent article with a group of people from Johns Hopkins on the genus Cannabis, remarkably important ethnobotanical and economic plant. Instead of summarizing the article myself, I will turn the program over to Dr. Russo.
Okay, well, thanks, Jim, and welcome, everyone. I'm going to share my screen and get the slideshow set up. Let's see. Hopefully everybody can see that. And I'll just do that. So here's our title. We're going to talk about limonene, one of the key terpenoids of cannabis, and THC, providing support for cannabis synergy and the entourage effect. And this is just the name of the company, Credo Science, a couple of molecules for those who are so inclined. This is going to be a little bit different than some journal clubs where they just review the article and answer questions. I've got a great deal of background information. I culled this down from 100 slides, believe it or not, but I'll provide a lot of background before we get to the article, and hopefully they'll have an understanding of how this issue arose historically and what basic science support there was for the clinical study that ensued. So this is a well-known problem with cannabis related to THC. When there's too much, whether it's smoked or taken orally, overdoses consist of panic reactions or what's called a toxic psychosis, which sounds ominous, but it is a self-limited condition. Generally, people do not need tranquilizers for it, but often can be talked down. And within a few hours, or certainly by the next day, there are no sequelae except that the person may have really scared themselves. So one of the interesting things is cannabis actually contains different compounds which can suppress THC-associated side effects. And first, let's get into the history of this a little bit. Back in 1998, Professors Meshulam and Ben Shabbat first described what they called the entourage effect. In the 1998 article, they were talking about molecules that were related to endogenous cannabinoids, specifically anandamide and 2-arachidonoglycerol, related structures that seemingly didn't have a lot of activity on their own, but which, when combined with the main endocannabinoids, produce synergistic effects. The next year, they extended this concept to cannabis with this quotation: "This type of synergism may play a role in the widely held but not experimentally based view that in some cases plants are better drugs than the natural products isolated from them." So this is a well-known issue in botanical medicine. For a while in France, there was one compound isolated from ginkgo biloba that was used to treat toxic shock, but it was found that extracts of the plant worked better. In relation to cannabis, this was a theory in 1999, but in the ensuing 25 years, we have a lot of evidence to support that what they said was true. One mention of this was about 1,500 years ago. Professor Lozano in Universidad de Granada in Spain had translated this from the original into Spanish, and I translated it into English. So this is a Persian Dr. Al-Razi. And to avoid these harms, meaning from too much cannabis or hashish, one should drink fresh water, chew ice, and eat any acid fruit. And of course, at that time, that would have been a lemon or a lime, particularly the latter in Persia. In the 13th century, Ibn al-Baitar said, "When one fears from some regrettable consequence, meaning cannabis, it is very useful to those who find themselves in this condition to take acid." Now, they don't mean that kind of acid. They, again, in this instance, meant citrus. And Ibn al-Baitar was in Moorish Spain in the 13th century. Bringing it to the West, William O'Shaughnessy was an Irish physician in service of the crown in Calcutta, in Bengal, India, in the 19th century. He said its use in accordance with the precepts of the ancient Eastern writers who prescribed hemp with alkaline substances and used acids in various forms to counteract its effects when taken in overdoses. He went back to England in 1843, and Indian hemp, so-called, quickly spread to Western Europe and North America. One of his acolytes was Alexander Christison. He had tried cannabis himself, had a bit of a lingering effect. Next day, he was perhaps more stupid than before, but was enlivened by drinking lemonade. And the next one, lemon juice, had a great effect in restoring my faculties. And then, as you see, lemon juice is stated by various authors to have great influence in modifying the action. It will arrest action entirely in many cases. So this is a continuing thread. In the United States, Fitzhugh Ludlow became famous for his experiments with medical extracts of cannabis. As lemon juice had sometimes an effectual cure for the sufferings of excess, he tried a bunch of other drugs too, but this was a spreading concept. And then this was in relation to opium and other drugs of so-called abuse. Again, deleterious as is hashish in the ordinary habitual use, it may be counteracted or neutralized very effectually for the time by the free use of lemon juice. And he made reference in Northern Africa The people of Tunis understand the secret thoroughly and avail themselves habitually of the benefits of lemon drinks. And then one of the great books on cannabis of the early 20th century was Walton. In the hangover period, which at times does follow the hashish experience, a great many have tried and recommended the administration of acid drinks. And just to demonstrate, in North Africa, this is an actual recipe for lemon water or lemon granita. And the key thing about it is it's not just lemon juice, but the zest of 7 lemons. And this is the source of limonene, which is the main component of all the citrus scents. And that would be a very strong dose of limonene when taken in this way. So it wasn't necessarily simple lemonade that they were drinking. Another reference was from Pliny the Elder in the 1st century, talking about galatophilus, which from Greek translates to leaves of laughter, growing in Bactria, which would be in current day Afghanistan. If this be taken in myrrh and wine, all kinds of phantoms beset the mind, meaning hallucinations, causing laughter which persists until the kernels of pine nuts are taken with pepper and honey and palm wine. So I investigated this, and it turns out that pine nuts are also rich in limonene as well as alpha-pinene. Alpha-pinene is an acetylcholinesterase inhibitor, and so will reduce short-term memory impairment induced by THC. The black pepper that's mentioned, its essential oil, also has alpha-pinene and as much as 16% limonene. So I was curious about what kind of foods might fit the bill, and looking at the content of pesto genovese. In fact, the high contents of limonene, alpha-pinene, and beta-pinene detected in pine nuts involve their significant contribution to the final months of these terpenes in pesto. So we have these listed. You see alpha-pinene and limonene again in the pesto genovese. So if we were to suggest a cannabis overdose antidote meal, it'd be great to have some pesto available with the agua limon on the side. The problem would be if you're already too high, it's going to be difficult to make these things. So this is why you need a handy chef. Some animal work from about 30 years ago, Buchbauer et al. So they exposed mice to terpenoid odors for an hour in ambient air. And if people are not familiar, terpenoids are the aromatic components of cannabis and other plants that often provide the scent as well as taste. Profound effects were noted on activity levels in the mice, even at very low serum levels. And there are direct pharmacological effects. Particularly here, we're interested in orange terpenes, which would be primarily limonene. There was a 35% increase in motility, even at a time when there was no detectable serum concentration of the material. The explanation be that this would rapidly enter the brain and no longer be detectable in the serum. An interesting study in humans was done in Japan in 1995. So they had 20 hospitalized patients with major depression. 8 just stayed on their normal antidepressants, and 12 were exposed to a citrus fragrance that was rich in limonene. After 4 to 11 weeks, the Hamilton rating scales for depression and rating depression scale were greatly reduced, and you see the reductions here. Additionally, the patients who were exposed to the citrus fragrance were able to reduce their antidepressant dosage, and 9 out of 12 discontinued that entirely. Whereas the patients who were only on antidepressants still required the usual dosing. There were also benefits on immune function. with reduction in cortisol and improvement in the CD4 and 8 cell counts. So it wasn't just an issue of mood being better, but there were, again, biochemical differences showing decreased stress. In a more recent study in 2021, it was noted that limonene is an agonist at the adenosine A2A receptor. Levels needed are probably pretty high, 5 micromolar. In this study, the mice were injected with limonene, and they looked at locomotor activity and open arm preference in the elevated plus maze, and that was statistically significant. There were also changes in dopamine levels in the striatum, and increase in GABA release in PC12 cells. The anxiolytic effect, reduction of anxiety, and GABA increase were blocked when an adenosine A2A antagonist was employed. Now, let's turn to the article finally. So as mentioned, this was done with colleagues at Johns Hopkins, and I should mention that this is a study that I've tried to do for more than 20 years. And we finally got the situation of getting an NIH grant. I like to say it took me 20 years to be a good old boy and recognized as an asset rather than a controversial figure with NIH. So this involved 20 healthy adult occasional cannabis users. The study involved vaporization of THC with or without limonene, and it was a double-blind randomized controlled trial. 12 of the participants also received a higher dose, and you'll see the doses in the following slides. So the ultimate dose is 30 milligrams of THC, a very high dose for vaporization, along with 15 milligrams of D-limonene. We'll go into more detail, but highlights the D-limonene reduced THC-induced anxiety and paranoia with a dose-dependent effect. It did this without altering THC-induced pharmacodynamic effects. And when taken alone, there were no discernible effects or adverse events associated. Part of the issue would be there might be different tastes associated if there's lemon scent versus not there. So part of the blinding involved a procedure where people would use a vaporizer, and then they would exhale through this device called a sploofie, which contains carbon filters and various filtrations so that what you exhale doesn't create any odor. And this is felt to be an effective method of improving the blinding. Here we look at some of the results, and you see that the doses range from 0 milligrams limonene to 1 milligram, 5 milligrams, and ultimately in the subset, 15. And doses of THC range from 0 to 15 to 30 milligrams. We have visual analog scale here, change from baseline for anxiety, paranoia, heart racing. So from the article, the main effect of treatment was observed for the anxious and nervous issues, highly statistically significant with a p-value of 0.01. Same for paranoid, and heart racing was actually 0.0001. Comparison showed that rating of anxious, nervous, and paranoia were significantly lower, particularly in the 30 milligram THC plus 15 milligram D-limonene condition compared to 30 milligrams of THC alone. And that, again, was statistically significant. None of the other planned comparisons were significantly different. On the state anxiety questionnaire, there was again a significant treatment effect, 0.01. Other didn't reach the threshold, but reductions in anxiety were apparent in a dose-orderly fashion. In other words, there was a dose-response curve that could be demonstrated at each, and it became most salient at the highest dose. Similarly, looking at drug effect, pleasant drug effect, and unpleasant drug effect with the different comparisons of D-limonene with or without THC. There was a mean effect of treatment for all three items that were statistically significant. Plan comparisons revealed that 30 milligrams THC plus 15 milligrams of D-limonene produced lower subjective ratings for unpleasant compared to 30 milligrams of THC alone. None of the other items were significant. This is a whole bunch of data looking at every kind of parameter that was tested on the drug effect questionnaire, things that are affected by THC exposure. And these increased or decreased depending on what the combination was for vaporization. Of note, the 1 milligram and 5 milligrams of D-limonene alone didn't produce any changes of subjective ratings compared to placebo. That's despite the fact that in aromatherapy, wafting limonene scent in the air often is associated with a subjective sense of improved mood. Here, though, we were testing for anxiety. On this, after drug administration, there was a significant mean effect of treatment for the digit symbol substitution task, 0.05, but not for the paste serial addition task. These are things that can induce anxiety. Plan comparisons didn't note any other significant differences in the combinations. We actually did pharmacokinetics here to make sure that there weren't alterations in plasma THC concentrations attributable to limonene that could help explain. Of course, there was a mean effect for THC with the 30 milligram of THC and 15 milligrams of limonene. There were greater concentrations of THC relative to 30 milligrams THC alone, but no other major differences. Now, this could have had something to do with differences in inhalation, but efforts were made to try and control this. There was a significant mean effect for treatment of concentrations of limonene, which was highly statistically significant. And again, there was a dose-dependent increase in D-limonene in a given THC group. When 5 milligrams of D-limonene was combined with 15 milligrams and 30 milligrams of THC, there was a significantly greater concentration of D-limonene relative to 5 milligrams of limonene alone. These are full-time course pharmacokinetic data for the D-limonene THC and its primary metabolite, 11-hydroxy-THC, which is what's produced after first passed in the liver and which is itself psychoactive. So for the pharmacologists in the audience, this may be of interest. And these are just from the discussion. I'll read. This experiment showed that simultaneously administering vaporized D-limonene and THC reduced subjective indices of THC-induced anxiety in a dose-orderly manner and corroborate historical references to ingestion of lemon or citrus juices as antidotes to overdose of cannabis or hashish. Again, this is reference to a review article, Taming THC, that I did in 2011. Again, quoting, in summary, the present controlled human laboratory study found that cannabis terpene D-limonene attenuated THC and induced anxiety in a dose-orderly fashion, but it had little impact on other common, acute, subjective, cognitive, or physiological effects of THC in the sample of healthy adults. Moreover, when inhaled alone, D-limonene did not produce any acute effects that differed from placebo. This is among the first clinical studies to demonstrate the validity of the cannabis entourage effect, which theorizes that THC and other constituents of the plant interact in meaningful ways that alter acute cannabis effects. Given the growing interest in the use of cannabis for medicinal purposes and expanding legalization of cannabis for non-medical purposes, further understanding of which constituents may increase safety profile of cannabis by attenuating acute adverse events, for example, anxiety and paranoia, and which constituents may exacerbate adverse effects, is paramount for advancing the use of cannabinoids in medicine and more broadly protecting public health. And I think that's it. So I hope I've left ample time for questions.
Thank you very much, Dr. Russo. That was very interesting and really a landmark study, one that directly addresses with all the proper clinical parameters exactly what effect limonene and THC have together. So thank you for that. And it sounds like it was a culmination of 20 years of effort. I had a question, which was with the potential rescheduling of cannabis from 1 to 3, will that make it easier for you in your trajectory in the future with similar studies?
Well, yes and no. First of all, it's hard to justify on a scientific basis. Really, cannabis doesn't fit in the schedules. I feel that it merits descheduling, so it could be legal and regulated, somewhat akin to the way that alcohol and tobacco are today, which are both substances that are obviously more dangerous than cannabis. That's not to say it can't be abused. I mean, we're talking about here of sequelae of excess THC. But the place where the scheduling makes a difference will be in clinical research. As it is now, as a Schedule 1 drug, it's a really onerous and convoluted process to try and do studies of this type. A Schedule 1 license requires approval from the Drug Enforcement Administration and often additional investigation and scrutiny from state authorities. It requires that the cannabis material be in a refrigerated, locked safe, in a locked room with limited access, subject to DEA inspection on the spur of the moment, without advance warning. And it's a very difficult and expensive process to do any studies of this type. So that would be easier. But Schedule 3 is designed for an FDA-approved pharmaceutical. It really isn't designed for cannabis. It would even be possible that in some jurisdictions that certain states or law enforcement would feel that this was a good opportunity for them to clamp down on existing commerce in cannabis. So there could be deleterious sequelae of the decision, but we'll need to see how it's put into effect. One thing's for sure, the FDA doesn't have the capability or the personnel to try and monitor this. And one reason this has taken so long to happen after more than 50 years of prohibition is that nobody knew what to do with it. Nobody wanted the responsibility. Anyway, that's a long-winded answer to an interesting question.
Yeah, there are definitely political repercussions and implications for any kind of a change. Of course, yes, decriminalization or legalization is certainly the best option. I also had a comment on
Well,
um,
Olga
land.
had a question. I'm wondering if we could answer that.
Yeah.
It's in the chat.
So
Ethan, do you-
Olga,
Oh, of course.
are there other
Yeah,
terpenes that
yes.
interact with THC? Well, I'm so glad you asked. Yes, many. And so I might refer you to the article I mentioned, Taming THC from 2011. So if you go to ethanrusso.org, at the library tab, you could put in Taming THC and you'd get access to that. It's all free. All my articles are there. So I'll put that in the chat.
And Olga and Katharina, since there's such a small group, please feel free to unmute yourselves and just ask questions.
Okay, but to answer
Thank
the
you.
question, let me just put this there. To answer Olga's questions, are there other terpenes that could interact with THC? Yes, many. Specifically at Johns Hopkins, we have follow-up studies to the limonene studies that would look at myrcene. Myrcene in THC produces what I like to call a couch lock effect. So myrcene has narcotic type qualities. It does produce sedation, and it actually produces analgesia that can be reversed by naloxone, the mu opioid blocker. So we're interested in whether we can demonstrate an effect that's been noted by many cannabis users, that high myrcene with high THC can be immobilizing. The other one that we're trying to study is alpha-pinene. I mentioned previously, this is an acetylcholinesterase inhibitor. Acetylcholine, of course, is a neurotransmitter and memory molecule in the brain. And one of the cardinal sequelae of THC excess is short-term memory impairment, which can be reduced or eliminated when there is alpha-pinene in the cannabis material. So we're going to test that as well. There have been negative studies done previously. However, these largely looked at the effects of cannabis terpenes, specifically at cannabinoid receptors. Now, interestingly, beta-caryophyllene is a prevalent terpenoid in cannabis. It is a CB2 agonist. So the cannabinoid 2 receptor is a non-psychoactive receptor that is anti-inflammatory and immunomodulatory without producing intoxication. Interestingly, it's selective for CB2 and has no activity at CB1. There are studies again underway with my colleague Ziva Cooper at UCLA to look at combination of THC and beta-caryophyllene to see if that affects pain thresholds, like in the cold presser test, where people dunk their arm in super cold water and see how long they can stand it. So yeah, many. And, again, I think that there's endless possibilities here in looking at some of these interactions, and it just doesn't need to be confined to THC. There could be combinations with cannabidiol, cannabigerol. We have a study out for publication now with scientific reports, double-blind, controlled field trial of cannabigerol in a pretty pure form, and showed prominent effects on anxiety. Now, combining that with limonene, or especially linalool, which is the main ingredient of lavender essential oil, with a well-known anti-anxiety effect without overt sedation, that would be a really interesting combination to see if there's synergy between those components. And Jim, you had another question?
I had one I wanted to ask, and it seems that I mean, it's well, you've had everything well characterized, etc. But I'm kind of wondering, when you're measuring things like anxiety or paranoia and stuff, how accurate are those, since they're so subjective?
Well, that's it. But, this is the reason we don't test in single individuals and say that it's significant. this was not a clinical anxiety population. However, certainly we have noted anecdotally that, again, various cannabis components can be very helpful in people that do identify anxiety as a clinical problem for them. And, it's very interesting that THC has a biphasic dose response with reference to anxiety. Tiny doses help anxiety. Too much obviously provokes anxiety. But again, historically, this was an effect that was noted by the Sumerians and Akkadians 4,000 years ago. There was a cuneiform tablet under a description of what we believe is cannabis. One of the attributes was for or against panic. So this biphasic dose response was noted by the ancients. We just have better tools now. So we're not making marks in clay and baking them to do our writing. We have PDFs on demand now.
Yes, we've come a long way, but they were important pioneers.
Sure. And the other thing I'd emphasize is I don't think that we've had, in 4,000 years of evolution, I don't think that humans are innately smarter than they were then. Again, science is advanced and we have better tools, but there was some tremendously astute observation going on long before these tools were available in modern science. That's why I pay attention to these historical yarns. Yes, Olga?
Question. So it says that the beta-limonene doesn't directly alter the function of THC and CB1 and CB2 receptors, but does the limonene reduce the medicinal effects of cannabis, or doesn't have any effects in the medicinal effects?
Well, again, what has been observed anecdotally is that this has a mood brightening effect. If people take THC alone, they find it quite disoriented and even dysphoric. Whereas with these other components, it definitely has a different flavor to the experience. And again, we don't know everything about how this works. The adenosine A2A receptor may be involved. Again, this is a hard thing to study, but we could look at things like PET scanning or functional MRI scanning to see what parts of the brain are affected by limonene with or without THC. So that would be an interesting follow-up study, but that gets extremely expensive.
Thank you.
Yeah, I was going to comment that from my readings, it appears that a lot of what people associate with the indica type of cannabis, as well as the more sedative, body-centered, relaxing effects from the indica type cannabis, quote unquote, is in sharp contrast to the more equatorial or sativa or cerebral or racy effects, which are actually often associated with the lemon flavor. So I just wonder if people over the millennia chose these racy plants, very strong with strong effects, but in conjunction with limonene in their selective breeding.
Right. Well, they may not have known what it was that they were doing, but yeah, the answer is yes. Most breeders do not breed for terpenoid content. However, it can be done. These are all Mendelian traits that without genetic modification, it is certainly possible and has been done to develop high limonene chemovars, chemical varieties of cannabis. where it can become the predominant and a high potency component. One of the criticisms we've had of this was that you would not likely find a type of cannabis where you got 30 milligrams of THC with 15 milligrams of limonene. However, there are many varieties of cannabis where there's little or no THC, and you can have significant amounts of limonene. So it is possible that you could get these kind of proportions. People had said that you can't, but actually we've seen that. So it's true to say that the cannabis of current commerce usually wouldn't fit these kind of parameters. But these things could be achieved, and certainly preparations could be made or blends could be made to produce any kind of ratio that you wished.
It brings to mind the modern THC vaporizer, which, for what I know, involves extracting THC from the other volatile compounds and then re-adding those in a way to try and recreate the overall, effect of the original strain. But if you put a little more limonene in it, it might be more comfortable for some people to smoke.
Right. So I'm not advocating spiking in that fashion. I should point out that although some of these components have different boiling points, it's not likely. Some people will vaporize at a very low temperature to get what they call the flavor hit, where there are more terpenoids, and then they turn up the temperature to volatilize more of the cannabinoids. But there can be a diminishing return, and you can get polyaromatic hydrocarbons if the temperature is too high, as it would be in smoking. It's not possible to isolate single components from cannabis through vaporization, because although we like to use, single figures for boiling points, a lot of these materials will sublimate off at a temperature that's far below the actual boiling point. So Trish would note this in Colorado that it could be 0 degrees Fahrenheit on a sunny day with wind where the snow blows into the air and disappears. It doesn't turn into rain. It just becomes vapor. So that is a sublimation effect. So the same thing can happen, and it's better to think about these boiling points as a range, and it may begin well below the actual boiling point that's cited in the literature.
Well, that may be it, unless there are any more questions.
No,
No, sure. Go ahead, Trish.
I just wanted to mention that we are having a meeting in June, the Society for Ethnobotany. If you're interested, it's in Michigan, and it's, I think, the 15th. I wrote it down someplace, but I don't have it in front of me now that I was taking notes. And so you could look that up under botanyconference.org or botany24.org and look further. And if you have comments about the journal club and you have some ideas either of speakers or articles that you would like to talk about or submit, we'd love to hear that. Yeah, didn't know if you had anything else you wanted to say, Jim, besides a great thank you to Ethan and Olga for attending, and Catherine had to go.
Sure. Well, thanks for the opportunity, and hope to see you all again sometime. Cheers.
Thank you. It was
Thank you.
interesting. Thank you.
You're welcome. Bye now.